Okay, why don't we go ahead and get started. Thank you all for coming. I'm actually going to make two introductions today. One is actually to tell you about a new activity of the Department of Medical Informatics and Clinical Epidemiology. And then my second introduction will be our guest of honor. So the first thing I'm going to tell you about is a new initiative in DMICE called the Informatics Discovery Lab. I know some of you know a little bit about it, but it represents our efforts in the department to reach out, to take on new work consistent with our mission that has a different audience besides government funders and the other usual sources. And just to quote, the mission for the Informatics Discovery Lab is to provide leadership, discovery, and dissemination of knowledge in clinical informatics, clinical epidemiology, and bioinformatics and computational biology. And we fulfill the mission through research, education, collaboration, and service. The IDL effort is being spearheaded by one of our faculty, Dr. Aaron Cohen, who many of you know. And part of this effort is bringing in some outside speakers who not only have subject expertise in aspects of informatics, but also experience in these sorts of activities. And so we've invited our guest today, Dr. Atul Butte from Stanford, who is here to not only meet with us to talk about our next steps with the IDL, but also to give a public talk and inform you of some very exciting work that he does that is, well, certainly if you look at what's going on at OHSU, is also the direction that OHSU is taking, which is the kind of integration of genomics and medicine and related areas, a very data-driven future. Dr. Butte is Associate Professor of Pediatrics at Stanford. He is Chief of the Division of Systems Medicine in the Department of Pediatrics. He also has numerous other appointments in departments around campus in genetics, computer science, and medicine. And he is initially trained in computer science and spent some time in industry, and then went to medical school at Brown University, where he graduated in 1995. He then did a residency and fellowship in pediatrics, fellowship in pediatric endocrinology at Children's Hospital in Boston, and then joined the informatics training program at Harvard, where he got a PhD in the Health Sciences and Technology program, the joint program between MIT and Harvard Medical School. He then moved on to Stanford, where he has led a very successful research program, and he is going to tell us about the work that he's doing. We were originally going to have this talk in our usual setting, which is the Bick Theater, which many of you know. But we were kicked out of there by the Bring Your Child to Work Day. But Atul actually tells me that that's actually quite relevant, and some of the things he's going to talk about deal with children. So I will turn it over to Atul and
Great.
enjoy his talk.
No, thank you very much. Thanks for the introduction. I'm just going to start with an apology. I've been in this field for 15 years. This is actually my first time finally visiting your campus. It's about time. So I humbly apologize for my delayed arrival here. First, it's a medical talk, so you always got to start with the disclosures. I started a bunch of companies. I consult for a bunch of companies. Now my average grad student starts a company. You shouldn't believe another word I say. But hopefully I'm going to convince you by the end of this talk to start a company as well, especially if you're a junior person here. All right, so if you haven't heard, we're in the middle of this data deluge. Let's just start with these deluges and tsunamis. Special issue of The Economist magazine 2 years ago. The cover was the data deluge, and in that issue it says that we as a human species now generate 2 zettabytes of data per year, 2 zettabytes. If you have forgotten your metric prefixes, it goes kilo, mega, giga, terabyte, peta, exa, zettabytes. And of course, next year, we as a human species will generate 4 zettabytes because it's doubling every year. And of course, the vast majority of this are random kittens playing piano videos. I grant you that not all of the zettabytes are useful, but they're entertaining perhaps. A large component of internet traffic is Netflix. I acknowledge all that. But there's a lot of data out there too, data that we can operate on in science. And there's data for particle physics. So Large Hadron Collider comes up with petabytes. We have the NASA Sky Survey, which takes such detailed pictures of the sky, not enough scientists even look at it. They crowdsource that to get the internet community to help look at these sky pictures, see what's out there. And we, in fact, have so much data now that's led people, including Chris Anderson, who was editor-in-chief of Wired, to write this article in Wired. Now, Wired is not the most scientific of journals, but this article intrigued me, that we collect so much data now. We collect so much data that science itself is growing obsolete, okay? And this is going to be a theme for the talk here. How can you say science is growing obsolete? Because remember the good old-fashioned scientific method. Scientific method is you come up with a question or a hypothesis, and then you go make measurements to address that question or address that hypothesis. And now we're in a world where we already have the measurements. We have so many measurements. The new magic I'm talking about is figuring out what's the question we want to ask. What do we want to do with this data? And once you figure that out, actually the code kind of even writes itself. But what do we want to ask that everyone wants to know the answer to, and nobody realizes is askable, and we have the tools to ask and answer today. So I'm going to show you this kind of approach of coming up with questions given the data. Obviously, it's not just particle physics and astronomy, it's in life science research as well. Another great article in Harvard Business Review. By the way, if you're a junior faculty starting a lab, I highly recommend reading the Harvard Business Review. It talks about how to deal with prima donnas. It tells about how to manage teams of innovators. It's a great magazine. Freaking expensive, but it's a great journal. You know, the next scientific revolution, moving from theory in the Greeks and the Chinese to experimentation in the 1700s, simulations and computers, and think about model organisms in the 1900s, now data mining in the 2000s. Nature says it's shameful if we don't use the data that's out there. Science says we need to make data maximally available, and even the Lancet says if we don't share research data, it's an impediment to public health. What kind of data am I talking about? A lot of it comes from high throughput measurements in molecular biology today. On the left here is just one example of such a technology. This is one source of all this data. On the left is just a standard Affymetrix gene chip. This is a chip that's the size of your thumbnail. Here's your thumbnail. Here's the chip. And this chip lets us quantitate every gene in the genome. We all know this. This quantities the RNA, the blueprints, not the DNA, not the protein, in between the RNA. And this lets us quantitate every gene in the genome on a single chip. Now, of course, we love to scale in life science. So when we want to scale this up, we make 96 wells of things, and each well here is one of these chips. So now we can measure them 96 at a time. Each well here is a chip that lets us digitally measure every gene in the genome. This is an amazing technology. You have to admit, it still looks kind of cool. By design, it kind of looks like an Intel microprocessor. They're based in Santa Clara, not too far from our Intel side of things in Silicon Valley. But it's amazing, and we got to remember, this today is a commodity item. Most people don't even care what company they're ordering from anymore. Affymetrix, Agilent, Illumina. It's only about $200 to $300 now for each one of these chips. In fact, it's been like this now. We've had these chips for 15 years already. We've had microarrays, 15 years. Everyone knows about microarrays. Everyone uses microarrays. And what happened in early 2000s is everyone started writing papers about microarrays, right? Here's a list of genes that are different in that cancer. Here's a list of genes that are different in this diabetes and this metabolic disease. And everyone started putting out, here's a list of genes, there's a list of genes. And what happened is the journals took leadership here, and the journals came out and said, hold on a second, guys. Guys and gals, no more papers on microarrays. No more, okay? That's it. Unless you start to deposit your data into an international repository. So other people can kind of double-check your math, and it's getting increasingly important to double-check some of that microarray math now that we're getting closer to the clinic. And maybe someday someone else can use the samples for something else. And the precedent for this goes back to GenBank, right, with DNA sequences. Nobody wants to type in DNA sequences from a paper. That's why GenBank is now 29 years old, okay? We've had GenBank for a long time. The same precedent happens for other microarray, other measurement technologies as well. Another kick, though, comes from these policies. So those of you who write grants and have to reapply for grants know that as we write papers, not with microarrays or otherwise, we have to make our papers open access now within a year. And we've always had this at NIH. The White House 2 months ago said, you know what, actually, now all funding agencies in the government have to make their papers available in a year, not just NIH. It was not a big deal for us. A lot of folks in our community said, oh, they're selling out. They should make these papers available on arrival. I am not going to argue any of that. One thing that people didn't catch in all of these White House announcements is that you got to make the data available too, not just the papers, okay? It's going to be a new world for us to think about raw research data getting out there. We have these precedents, but you're going to see more of it. So everyone knows what a microarray is, and most people know you have to share that data when you write a paper. How much data is actually publicly available today? And in last August, so August 1st, Nature did a special article. It featured my lab, a couple others. As of August 1st last year, we hit 1 million publicly available microarrays now. 1 million of them. Here's the exponential growth trajectory. Kind of looks like it's flattening off here, only because they counted half the year. Okay? We're still doubling every 2 to 3 years how much microarray data is out there. And to kind of make it really kind of obvious, if you go to the NCBI, we all know PubMed, some of us know GenBank. Well, here's a repository for this kind of data, NCBI Geo, Gene Expression Omnibus. Almost 900,000 arrays there right now, if you look. This is a couple weeks old. The Europeans run a repository called Array Express. They got another 250,000. Actually, today we're already blown past 1.1 million. Right? 1 million was all the way back in August. That's eons ago. We're already got another 100,000 arrays here. Now, let me just make it painfully obvious what this means to all of us in this audience. In the middle of this website is a simple search box. Right? Some of you have heard me say this in talks. Maybe you haven't. Even a high school kid today, maybe who's here for back to see your parents at work day, right? A high school kid today, it needs to do a science fair project, right? She can type breast cancer here and now digitally download 34,000 digital samples of breast cancer about as easily as she can find a song on iTunes. Okay? 1,300 plus independent experiments here contributing this data. Now, let's just kind of make this obvious. If it's not obvious to you, 34,000 samples, why is that interesting? Because that's more samples of breast cancer than any breast cancer researcher will ever have in their lab. Because every one lab has to share their data plus their competitors. So whoever figures out how to get to the middle is going to have more samples now than anyone else. Okay? Especially as most of the contributing people don't know how to get data out of this system. They only know how to put data in. And let me just point out the 1,300 labs. Let me just reverse this for a second here. Right? Let's say I came to you with an amazing grant proposal. I'm going to do this amazing grant where I'm going to get 1,300 of the best breast cancer researchers in the world. I'm going to ask them all to do all the experiments they're great at doing at studying breast cancer. Maybe it's cell lines or mouse models or human samples. And I'm going to get all 1,300 of my best friends, the best people in the world, to run microarrays, to share the data with me. And actually, I'm not even going to pay them $1 on my grant, right? So kids today would call that crowdsourcing, right? In the really old days, that was like Tom Sawyer, right? Getting your friends to paint the fence, right? I mean, now they call it crowdsourcing. So imagine crowdsourcing to the 1,300 best breast cancer labs, not paying them $1 and getting all their data. You'd laugh me out of the room if I proposed that. Yet what I'm doing here is retroactively crowdsourcing. The crowd is helping me with their best research findings, and they don't even know they're helping me, and I'm not even the one hitting them with the stick making them share this data. And nobody's really using this data at all today. Now, if you have any doubts, a high school kid can do this. I'm most proud of my 5 high school kids during their summers that have either won the Intel, Westinghouse, or Siemens semifinalists, or even made it all the way to the finals. He was #2 science kid in the country. Each one of these kids was one of the top 300 kids in science in high school of their respective years. A high school kid can absolutely do magic with 10,000, 20,000 samples of breast cancer, prostate cancer, whatever disease you want. The data is sitting there waiting for someone to analyze it and ask a question. Now, this is on the RNA side. You know, the DNA side is just the same way. You have to share data to dbGaP. The rules are a little bit more severe. You had to get permission sometimes, but the data is being shared across the board. Oh, I'm not even counting in this the Cancer Genome Atlas. Beautiful set of currently 18,000 cancer samples, including, yes, another 860 breast cancer samples if you're interested in breast cancer. But of course here they don't just have the RNA, they have the RNA-seq, they have the DNA changes in the oncogenes, they have the methylation, they have copy number changes in the DNA, and the histopath images and the mammography, okay? Everything from the diagnostic radiology to the microarray and all the different molecular measurements, and they give this data away. Imagine how you can be the person coming in, connecting the imaging to the molecular side, right? Things that each one of these cancers is written in a big Nature paper. I grant you, they get their Nature paper, and then they move on to the next one, right? And all that data is sitting there, no one really uses it for much. And it's up to you to think about what you want to do with this. If we're talking on the drug side here, because I know we have some drug discussions coming up later, here's another repository even fewer people use, called PubChem. Just show of hands, how many people have heard of PubChem? Oh my goodness, 3-4 people max. PubChem is another repository. What happened in the roadmap initiative from Elias Zerhouni said, we need to get academics starting to think about drug development, not to make them full pharma, but at least start to pay for screening tests. And these are screening tools where maybe you have a cancer that glows if it's living or glows if it's dying. You put them in wells, you test different drugs with robots, and these are screening assays. But NIH said, if we're funding you to do a screening assay or to develop a screening assay, you got to dump your data into PubChem. Fast forward 10 years later, what is PubChem? Think of a grid of 100 million chemicals as the columns, many of which are drugs or future drugs, 650,000 screening assays, bioassays of the rows. If you do the math there, that's about 70 trillion little boxes, of which 1 billion of them have some poor scientist somewhere made a measurement and stuck it in this whole system. Now, I'm willing to bet any one of you, this doesn't probably just beat the screening database of a pharmaceutical company. I'm willing to bet this probably beats them all combined. This is more data that's publicly available, I'm willing to bet you, than all pharmaceutical companies combined, because this is doubling every 2 to 3 years again. And nothing in pharma discovery is doubling at that rate anymore. And they give this to you without a user ID and password. Just sitting there are all these drugs, chemical properties, waiting for something to do something magical with. Of course, I got to be really a big fan of this one, Import. This one I took over in January, a $30 million grant, basically from the NIAID, Allergy Infectious Disease. They have their stick that says if you're funded by NIAID, you got to share your data in a repository. Now, this has a lot of data that doesn't have any other home, like flow cytometry data, CyTOF, novel ways to sort cells and to look at their cell properties. And that's great, and this is another repository. I've had this grant for 90 days. And what I realize this data is going to be about is just completely change my perspective on everything. The best thing that's going to come out of Import and maybe other repositories in the future, the best thing is going to be raw clinical trials data. This is going to be the next big revolution for us, okay? I don't mean the summary statistics that you're mandated to put into clinicaltrials.gov. I mean, imagine having individual patient-level process, like how did this patient do in this trial? Which arm were they in? What were their outcomes? Imagine being able to reanalyze and meta-analyzing trials without being a part of those trials. Imagine the future app of the future where a patient can say, boy, I most closely resemble this patient in this trial. What happened to this one and that one? Those are things we can't even imagine today that are going to be possible when we get raw trials data. And so this is going to be one of the first repositories. We already have 3 trials of data out there waiting for you to play with. Now, this is great. We can start to get to this data. We can start to turn our computational crank. We got our methodology, and we can start to get ideas. We can get ideas like, this is a great drug that might work on this disease, or, here's a good blood test for this particular disease to tell me who's going to get this disease. And then all of a sudden, we hit a wall. How do we as computational folks convince our basic biology friends, our clinical scientist friends to test these ideas? Because we can come up with ideas, 100 a day, 100 an hour, and sometimes it's lost on folks why they should actually pursue these to test them in the lab. And we all have good friends and collaborators. You have great cancer centers here, and we have them too. But at a certain point, you have ideas, and you're not able to find people to test these ideas. And then what I'm going to show you in the next few slides is going to be perhaps even more eye-opening to you. So the other day, we had this prediction. We used some of our own data, some public data. We came up with a good serum protein test for acute myeloid leukemia. Not a good leukemia to have, perhaps one of the better ones where you talk about having a bone marrow transplantation. Maybe this would be a good test to see who's going to relapse or not. But we go to our cancer center, we're NCI funded. We say, do we have any saved serum or plasma on patients with acute myeloid leukemia? And we're NCI designated, we have a biorepository. But it turns out they saved all the cancer cells. They saved all the cells and the RNA from these cancers. And that's great, but they never saved the liquid around the cells, the serum or the plasma. And it's a strategic decision. It takes more freezers. You got to save them all. They say, that's okay. Go get IRB approval. Go put up the posters. We all have posters recruiting patients. And you should have enough patients in a year to test your computational prediction. And what does a computational guy like me do? We just Google for it. And we find companies like this. Conversant Bio. Browse by disease. I see pictures of vacutainers. What do I want to buy today? Bladder cancer, brain cancer, breast cancer? Okay, here's leukemia. There's lupus, rheumatoid arthritis. What exactly do I want to purchase? CD19 positive B cells from bone marrow, CD45 positive cells from bone marrow, mononuclear cells from bone marrow, T cells negative selection from bone marrow. Page 2. Here's plasma from peripheral blood from leukemia. $55 each. How many samples do you want per patient? What disease subtype? How many units? Add to cart. What is this? This stuff shows up in 48 hours on dry ice, ready for you to go. What is this? These are huge hospitals in the middle of the country that get blood tests on their patients, right? And you probably know, when a doctor or nurse orders a blood test, that blood sample sits in a freezer for 48 hours, waiting for them to add something more to the test. And at the end of 48 hours, to make room, that sample has to get thrown out. It gets incinerated. Legally, something magically happens when it leaves the freezer on the way to the trash. That is now called a discarded sample. And legally, you can strip off the identifiers, put on some basic clinical characteristics. We as customers have to agree we're not going to try to re-identify these people. I don't really care who these people are. But then we can purchase, and those companies organize these samples, resell them for research use. And you get these in 48 hours. Now, why am I chuckling at this? This is way faster and cheaper than my own institution can deliver these samples, right? So I'm not against my institution having a biobank, but it better be faster, cheaper, better than what I can get off the internet today, okay? $55, we just order them all, and we validate our markers this way. And you know those core facilities on campus? Nobody ever said we in informatics can't use them either. I don't want this stuff to show up to my offices and cubicles. I want it to go directly to the core facility. We're having a fire now. My remarks weren't that incendiary yet, but they'll get there. It's amazing that the alarms aren't going off in this room. So that's perhaps a blessing and a curse as we burn down. Okay, so pathology samples, I could tell you, you can buy tissue microarrays. You can buy pathology samples for a couple $100. Like, if you have questions, I can go into details. But then the ultimate, okay? We have this great data. We have our tools. We come up with ideas for drugs, right? Maybe we think this drug might work on this disease or this drug might work on that condition. How do you test that? Well, we can go run a trial, or we can go find our friends in an animal facility, or we can just go to assaydepot.com, okay? The folks that are chuckling recognize this sounds like Home Depot, okay, for good reason. What do you want to buy today? Biology, chemistry, drug metabolism, pharmacokinetics. Let's click on pharmacology. What type of mouse do you want to run today? Bone, cardiovascular, dermatology, diabetes, urinary, infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. I'm a diabetes guy. Let's click on diabetes. Okay, here's a standard 16 mouse experiment, OB mouse model. You have to know this mouse has been eating a lot and getting diabetes since 1951. Kind of teaches you that. This is one of the standard models to look at obesity-induced diabetes. And now if you have an idea for a drug, you can order the drug from one catalog, the mouse from this website, and you have 2 groups of 8 or 4 groups of 4. Over 28 days, they'll measure the insulin and the blood sugar levels, and they email you back all the experimental data. What am I covering up with yellow? The price. $9,000 for this service, 9-week turnaround time, and now literally add to shopping cart. That entire mouse experiment, whether you want primate, mouse, rats, the entire animal experiment is purchasable with e-commerce today. Okay? You can buy the entire mouse experiment with a decent-sized credit card. We do this like crazy in my lab. Now, some of you with sharp eyes have noticed this one's run in China. And some of you may have a problem with that, some of you not. But actually, in reality, when you hit add to shopping cart, you're actually presented with 133 companies that know how to offer this to you, including Maryland, Wisconsin, and places all over the world. So outsourcing doesn't mean offshoring, okay? You can have the place be wherever you want it to be. But actually, maybe you only want to see the USDA-approved companies. There's 4, FDA, 5, ISO 9001, 7, AAA-LAC, 28. You click on the certifications here, you're only going to see those companies with those certifications. Oh, how I wish I could pick my collaborators on campus this way, knowing how certified they really were. They're putting some random grad student on this thing, right? Now, some of you are dying to ask me at this point, what about the quality of the results here, right? The mouse, the drug, this is probably the most important part of your experiment here. How do you really trust this company to give you good quality results? And I'll simply answer that if you truly care about the answers from this type of company or the results you get back, simply order 2 of them. One from the East Coast, one from the West Coast, right? That's what we do. You're going to see examples of that. Why am I being so flippant about this? Because think about the random biologist that runs a mouse experiment. They get it working in their lab. Now, how many of them go across the hall and say, can you repeat my whole experiment without me to see if this works in your lab? Nobody does that. So now quality for me is a function of dollars. If I want really the best to know if this drug works, I'll spend more money on this, right? If I just need to see if it works, I'll order 1 of them. $9,000? Order 5 of them. If you really want to know if this drug is working, you just spend more money, get more independent labs doing this experiment. So I will always win the quality argument now, okay? Because it's just a matter of getting more dollars here. That's kind of the trick here. Now, the few basic scientists that are in the room here, the basic biologists here might say, you know, you look at this price, $9,000 for only 16 mice? That's incredibly expensive, okay? And that's true because those basic biologists have slaves. I mean, graduate students. They actually could do this work for much cheaper than $9,000, right? We think about us as computational dry bench researchers, right? If you visit my lab, it's all office and cubicles. The wettest thing we have is the coffee machine, okay? Office and cubicles, for us to get a mouse thing done is impossible. And so this is priceless for us to get this kind of data here. So let's fast forward to the bench-to-bedside problem, actually, right? You think about the translational pipeline, how we make clinical and molecular measurements, increase with sequencing or microarrays. We ask the good question or trial. We run our statistical computational methods to analyze the data with this question in mind. We then validate it in the second phase, let's say a drug or a biomarker. I'm just simply going to say today in this pipeline, 3 out of these 4 are commodity items or services today. We're sitting on 1 million microarrays just on the RNA side. We have plenty of computational methods. Look, I'm a computational guy, but I have to admit, our field just keeps churning out more and more methods, and it's not really clear why someone else would hit our website now compared to someone else's. There are plenty of websites. Okay? If you really built a great killer website, show people what you find with it, not just that it's a website. I mean, you're going to see that theme towards the end. And now I can validate that second phase, a drug or biomarker with a credit card to buy samples to get this done. So let's take this to an extreme. In my part of the country, in Silicon Valley, we're so used to starting huge companies in garages, right? Apple and HP are the famous ones. We start companies in dorm rooms, Google, Yahoo. Of course, Facebook starts in a dorm room out in Harvard. I really, truly believe the next Genentech, the next Amgen is going to start in your garage. Your next big pharmaceutical company is going to be in your garage, right? I'm not talking about making meth, okay, in your garage, okay? I'm talking about an Amgen or a Genentech, the next big biotech, what I'm calling garage biotechs. When you go home today and you look at your garage, you already have 1 million microarrays and every mouse model in the world available from your garage. What else do you need to get started with a biotech company, right? Of course, there's a lot of hard work after that. But what else do you need to get started here? There's a little bit of investment, that equals a decent-sized credit card. You got all the data and computational chops and get a little bit of brain power to know what to do next here. So I'm just going to give you anecdotes of how we've been doing this here. And so the first example I'm going to talk about is to go right after this kind of big picture, going after public health menaces. In my lab, we love going after public health menaces. This paper came out in PNAS last summer. It's a little bit more molecular, and then we'll talk about some clinical stuff. Type 2 diabetes is a public health menace. 1/3 of all kids in the United States are going to get type 2 diabetes. If they were born after the year 2000, there's a problem where the insulin doesn't work as well in the body. We've known about diabetes forever, for at least 100 years. We've known about insulin. We still come up with new drugs all the time. We have this new category of drugs that came out in 2008, DPP-4 inhibitors. I'm just giving this as an example drug. We've had drugs for 50 years, 60 years. But even new drugs show up on the market, and the first-year sales are immediately $1 billion. It's already a $6 billion market. That's how badly we still need new drugs in diabetes. Okay, I'm going to focus on diabetes for the next few slides. And I'm going to, and like everyone else, we had a microarray paper. This was back when I was at Harvard, almost 10 years ago. Seems like 100 years ago. Here's diabetic muscle, here's non-diabetic muscle. Run them on microarrays, 100 genes, different, big deal. Okay, everyone had a paper like this. Now, one paper is not going to be enough. And what I'm going to do is if you have any kind of interest in this kind of way of doing science, just remember this paper. Nature Methods, April 2009. I'm going to explain this paper because nobody really recognized this paper. This paper says questioning standardization of science. It comes from the mouse psychiatrists. Think about those two words together, okay? The mouse psychiatrists, those are folks who wrote this paper. And you can imagine people use mouse models for anxiety and depression. And all these folks were saying, boy, man, well, when we run an experiment at Stanford, we get these genes. And when they run an experiment at Harvard, they get those genes, and these genes are not matching. We're not getting reproducible results in mouse psychiatry. And I said, this is ridiculous. We need to standardize how we study mouse psychiatry. This is what it means to have anxiety. This is what it means to have depression. And you got to put the mouse in this maze and do this with that. And now, all of a sudden, everyone's getting the same answers, as you would imagine. That's what standards do. The Harvard lab, the Stanford lab, everyone's getting the same answers. These are the genes that are showing up. And then a problem happens. The minute they change one parameter in the standard way of doing the experiment, this kind of mouse to that kind of mouse, 1 mouse per cage, 2 mice per cage, this diet, that diet, give them something to play with, give them water and less water. The minute you change one aspect of the standard protocol, none of the answers hold up. They're not generalizable. So we in the computational world know what they did. They overfit their model. They said, if you do the experiment exactly this way, of course you get the same answer. But the answer is actually irrelevant if you just change one parameter there. This controversial paper came out and said, why on earth do we keep insisting we standardize things in science? We should deliberately try 100 ways to do this experiment and then figure out what's in common in the end, right? What sticks out at the end? Maybe not everyone gets the same answer, but there's stuff that's in common in the end. This paper totally resonated with me because we can do this in silico with the world's data. And so now I'm not so uptight about did they do the same experiment here. I just want everyone to do the best experiment they know how to do in diabetes and then figure out what's in common. That's the model here. So fast forward to today, instead of one random microarray paper, I look on the internet, I find 130 papers in this field, 130 experiments. I don't even need to write a single author. I can get their data off the internet, where everyone's looked at diabetes and control, diabetes and control, diabetes and control. But they might have looked at liver, muscle, fat, beta cells, the cells that make the insulin, rat, mouse, or human. 3 species, 4 tissues. Simple question. What's in common? Even a high school kid, I can prove to you, even a high school kid can do this. What's in common across all these models? Well, let's just look at the background. Here's a grid that goes from 0, going too fast, 0 to about 130 or so would be out here. These are just 25,000 genes in the genome. It's kind of not hard. Almost 25% of the genes in the genome show up in one person's experiment. Okay? That's how much noise there is here. A lot of genes show up. It gets harder, though, to show up in 20 experiments, 40 experiments, 60 experiments. You can kind of see a tail dropping off here. We got many known genes for type 2 diabetes, including PPAR gamma, drug target, leptin receptor, those OB mice, TCF72, GWAS hits. All I'm trying to show here is that the known genes show up shifted over to the right compared to all the rest of the genes in the genome. Okay, and you can put a p-value in this. I'm not even going to scare you away with the p-value. So we're looking at this, we're looking at this, we're looking at this, we're just simply noticing, what's this tiny little red dot out here? This is a gene, I'm going to call it gene A for the moment, that shows up in 78 experiments, and it's in more people's experiments than any other gene in the genome. And it's never been chased down for diabetes. Okay? And we look at gene A, gene A, gene A. It's got an interesting symbol. It's a CD molecule. Okay? Like CD4, CD8, maybe clinicians have heard of those too. That means that it's a gene that codes for a protein that sits on a cell surface, and some immunologists at one point in history could sort cells based on it. You could sort cells based on this surface protein. And then we're staring at it, we're realizing it's not just a cell surface protein, it's a functional receptor. So that means other things bind to it and turn things on and off in the cell. And in fact, the ligand, the thing that binds to the receptor, is gene B, and that's this red dot here. So we got sitting in everyone's data a brand new receptor for type 2 diabetes, okay? So we got to start chasing this down. And we enter this great collaboration with the University of Tokyo and 4 other institutions in Japan, Riken, Keio University, Kitasato University, Kitasato Medical School, and we had a lot of folks helping us with the mice. But where we didn't have someone helping us with the mice, you know how I'm getting the mice stuff done now, okay? So where do we even start? Well, let's go ask the database. The database says, well, it chased this thing down in the fat. The fat really seems to change this receptor a lot. And in fact, 90% of the fat experiments where the receptor changed, the ligand also changed. So it's probably what we call an autocrine effect. So now let's show you fat pictures. You can see how we can get this stuff done with collaborators, with just buying this. Here's this normal fat, high fat diet. Just take fat. The gene does go up by RNA. Stain, you get pretty pictures of fat. And you realize the receptors in these inflammatory cells. It's not a fat cell problem. The fat has inflammatory cells or immune cells in there. And that's what's making these receptors. They happen to be macrophages, so you happen to know anything about immunology. Ligand and receptor are co-expressed as we predicted here. The very next step the biologist would do is say, wow, this is kind of interesting. Maybe we got to see what this thing does. Let's go make a knockout mouse. Let's go make a mouse that's missing this receptor. And lo and behold, you look in the Jackson Labs website, the Jackson Labs where all good knockout mice go to live and die. Jackson Labs website, some immunologists knocked out this receptor 11 years ago. Mouse is at Jackson Labs, and they never looked at the sugar level of the mouse. Okay, you have to fast the mouse. Got to know how to do that. Nobody looked at the fasting blood sugar for this mouse. $6,000 later, we have the mouse. Okay, let's look at the mouse. Stain for macrophages, fewer of them. This quantitates it. You can see that by eye. But how does it do functionally? It turns out this mouse is insulin sensitive. Let me tell you what that means. You knock out this receptor, it doesn't die from diabetes. It does better than the control mouse, okay? It's more insulin sensitive, whether it's normal fat, high fat diet, fasting blood sugar, glucose test, insulin test. The mouse is insulin sensitive at the same weight, so it's not a weight issue here. Now that tells us, boy, if we knock out this receptor, maybe that could be a great therapeutic target someday for humans, for diabetes. I mean, it's a brand new receptor here, and knocking it out seems to do better. Well, let's go back to humans. Let's see if humans have this. How do we test human fat? Oh, can I tell you how easy it is to buy human fat off the internet? It's called liposuction. 57-year-old woman, BMI 37. You can guess where this fat came from. We don't know who this person is. We don't care. Loaded with cells with this receptor, okay? It's not even subtle. In fact, humans make so much of this receptor. There's a soluble form. It disconnects from the cells. It just floats in the blood. And you can buy that ELISA kit for $500. It turns out the level in the blood correlates with something called the hemoglobin A1C. That is the blood test we run on diabetics to see what their sugar levels are. And again, lower is better, lower is better. So $500 ELISA kit. These 55 folks, by the way, don't even have diabetes yet. So as it's going up, the A1C is going up, or vice versa, but the mouse tells us it's probably causal that way. And it actually has to do with insulin resistance. If you're really diabetes aficionados, you know what a HOMA-IR is. So everything is telling us, get rid of this receptor, knock it out or hit it, it's going to make a great drug. We said, oh man, let's just go right to the punchline. Let's make a brand new drug. So what's the simplest drug you can make? Well, here's a receptor. Here's an anti-receptor antibody, okay? Let's just treat mice with anti-receptor antibody, and the control gets isotype control other antibodies, random other antibodies. In about a week, we can wipe out that infiltrate, not 100%, maybe some T cells there, who knows? And we can lower the blood sugar in 7 days. So now let's recap. We've got a brand new prototype drug for type 2 diabetes, currently affecting 150 million worldwide. A third of all kids in the United States will get this disease. Last new drug hit $1 billion first-year sales. We've got a new prototype drug, new mechanism of action of getting disease, new mechanism of therapy to treat it. We've got the knockout mice, immunohistochemistry. Remember that blood test? Company at Genentech might call that a serum companion diagnostic, so we know when it's high. We can even watch it perhaps go down. And we started with the same data any high school kid can get to today. Okay? That's the story here. Right? The data sitting there, by definition, means 78 people missed this receptor, because when people run experiments, they love to see what they know. And they probably say, that's just inflammatory schmutz in there, right? Probably causal here, right? That's the thing, is it probably has more to do with just being the noise here. We've done this, by the way, what is the receptor? It's called CD44, the hyaluronic acid receptor. Those of you who are breast cancer researchers, maybe in the audience, know that we actually have anti-CD44 in development, because it's all of a sudden back in the press as being a cancer stem cell marker of all things. Now, all I'm going to say is it's a complicated receptor. A lot of exons alternatively spliced. Maybe the receptor in breast cancer is different than the diabetes tissues. Who knows? We'll see how this drug does. But it's a complicated receptor. We can do all sorts of fun biology. I'm not going to be the one to do the biology. I'm a computational person. I'm an informatics person. I get the launch here. We'll get drugs spun out of this thing, and we're off to the next disease. Okay? But we're going to launch biology. But we're starting from a huge point here. That's the way to do this. Now, we start to realize instead of doing this just for one disease, like a diabetes, why don't we just start to do it for all the different diseases? And so actually my first big NIH grant was just go get every human disease that's been studied by microarrays. That's what NIH CMS just renewed my R01 for. Go get them all. Fast forward many papers later, we text mine to find these things. We eyeball them to make sure we know what we're getting. That's in Nature Biotech. We know how to map AFI, adjuvant arrays, to the latest gene identifiers. That's in Nature Methods. We know how to deconvolve complex multicellular samples to single cell samples. That's in Nature Methods. All the papers, I'm not going to talk about any of the methodology. It tends to get really boring. I'm going to show you what we're doing with this. Fast forward, now you have a big grid. 300 plus diseases this way, 20,000 genes this way, and we know how much every gene is changing in every experiment, okay? Including leprosy, Marfan's, asthma, salmonella. These are all diseases people have studied. For the aficionados in the audience, we text mine. We actually started using MetaMap. Now we use something called from the NCBO, the Center for Biomedical Ontology. We code everything with SNOMED and represent it in UMLS. We love UMLS in the lab. It's just the best way to represent these things because you can join with ICD-9, ICD-10. And SNOMED is what the pathologists have been using for 70 plus years. It's probably going to make the basis of ICD-11, so we SNOMED code everything. For statistical folks in the audience, we rank, normalize, and scale every measurement. So we destroy any distribution that might have been in the array. Lowest gene on chip gets a 0, highest gene on chip gets a 1, and we have equally spaced fractions in between. So it's fairly brain dead types of, and we have methods to show that actually works the best for us. Now, while we're building the disease side, it turns out huge labs like the Broad Institute have been doing the same thing on the drug side. So Justin Lab and Todd Golub came up with something called the connectivity map, where they launched with 150 drugs. First was like 150 drugs, and they had 1,000 drugs, and now they're launching something called LINKS, which has 5,000 drugs in it. They take a whole bunch of cancer cells and say, what is the microarray? What does the gene expression look? With drug, without drug, with drug, without drug, with drug, without drug, with drug at this dose, and that dose, without drug. They tried different doses, many different drugs, and we start to realize we can partner these two public data sets together. And I'll make the statistics super simple with my arms. If I've got a disease where I got this gene going up and this gene going down, and now I've got a drug that makes this gene go down and this gene go up, maybe there's a match there, okay? And that's 2 genes, imagine 20,000 genes. And imagine a statistic, a negative correlation here. Now, we're not brain dead here. We know not every drug goes to, for example, the brain, blood-brain barrier. So you got to look at a lot of non-computational things to know what to pursue here. But boy, what we start to realize is that not only can we do this with just random drugs, we can start to find new uses for drugs. Drug repositioning, or what's called drug repurposing. It's got a great history of finding new uses for drugs. Some of you know there's a famous angina drug that had an annoying side effect of hair growth. That is today what we have, minoxidil. People know that was repositioned before it actually really even came out. It was in the trials. But of course, the classic example everyone knows about is another interesting cardiac drug, very interesting side effect. Patients don't want to give the drug back after the end of the trial, equals Viagra today. Now, everyone knows the Viagra story. I think very few people realize that Viagra's double done this, right? We have another drug called Revatio, same molecule, but supposedly indicated for pulmonary hypertension. It opens those blood vessels too, from the heart to the lungs, okay? But you know what? Instead of finding these by accident, why don't we try to find them on purpose using public big data here and these computational methods? So how are we going to do this? Wow, I can find a drug, and here's a drug for heart attack, and it seems like it's going to work in muscular dystrophy. Maybe I should just try a drug for this disease and try it in a kid with, even before I put it in a kid. I'm just going to put it in a mouse model for a couple $1,000s, right? That's how it's done. Why? I'm going to skip through this part. All right, why are we going to do this, and what are we going to do? First, turn our crank. First story we get, epilepsy drug, topiramate, called Topamax. It's a J&J drug, now off patent, available in generic. We think works on Crohn's disease, inflammatory bowel disease, so from the brain to the bowel. And we make a kind of grid like this. This is truncated here. And these are all different drugs. And we see steroids look like they're going to reverse the effect of inflammatory bowel disease. We know that one works. We treat patients with steroids all the time. We don't like it, can't stay on it long term. But better than steroids seems to be topiramate. Would make an amazing scientific story if this one works. So it's a no-brainer we're going to test it. It seems better than steroids. And those of you can just see this is just to eyeball it, right? Here's Crohn's disease. These genes in general are up, these are down, and here's topiramate, here's down, here's up. So I'm just trying to eyeball it that we're reversing the effect if you look across the genome. So we start to work with collaborators on campus. We start with Jay Pashreek, Mohan Shenoy, GI folks. And the way to do this is you get a rat. We have to learn. I'm not a GI expert. We're going to learn how to do this. Read some papers. Turns out you get rats, you squirt TNBS up where the sun don't shine, and you start to get inflammation in the bowels. Treat with steroids, it seems to reduce that effect. There's still some infiltrate. Treat with topiramate, it seems to reverse the effect. Now, we love these guys, Mohan and Jay. We submit this to Science Translational Medicine. The editors come back and say, guys, do you have any better data on this one? I love those two, but they were giving us iPhone pictures of the colon, okay? Not the highest resolution camera. I love these guys. We knew the drug was working, but not the best data. So they need better data. Editors are asking for better data. We said, okay, Jay, we love you. We're going to outsource this one. Assay Depot, 100 companies come back to us in 24 hours. We pick one out of Berkeley, kind of because they're having a sale. For the same price as everyone else, they're getting us all these serum cytokines, inflammatory markers, something called Luminex bead assay. So we get TNF alpha levels, kind of useful for this. So, okay, we're going with you guys. And then we pick a company out in Cambridge, Massachusetts. Freaking expensive, but the only ones in the world who knew how to do a rat colonoscopy. This is a normal rat colon. This is a rat with Crohn's disease or inflammatory bowel disease with the TNBS. You can see it here. And this is a rat with TNBS, now with the topiramate. Now you know I'm not just cherry picking the images for you. I have 40 megs of video with the paper. You go look to see what the colon looks like here. Now, why is this important? It's an important point to realize, as a computational person, I'm not just outsourcing this stuff to save money. I'm outsourcing this to get the best damn data in the world. Nobody on campus knew how to do a rat colonoscopy. Those of you kind of chuckling maybe are over age 50 and you've done the geometry here. This is a colonoscope and this is a rat, okay? So you got to really have some special skills to know how to do this, okay? This is exactly the kind of experiment you want to outsource, okay? Someone else can figure out how to do this. So we do this all the time now. 2 companies, they give us the same answers, plus our own on-campus collaborators. We really know this drug is working. And out goes a company, spin-out company that turns the crank computationally as a business model here. We got working for other cancers. I'm going to show you this one here, okay? This is one I'm most proud of. We've done 6 of these. 6 out of 6 have worked in animal models for us, okay? 4 more papers coming out, one by one by one. This one I'm really proud of. This is a drug. This is, first of all, a mouse that gets its own lung cancer. If you knock out P53, RBP130, it's a triple knockout. It gets its own small cell lung cancer. So you got to wait weeks for it to get cancer, but you can see nodules here. Your projector is great. The lighting is perfect. You can see nodules here. And this is a mouse, same mouse, treated with an antidepressant. Still in the pharmacy, but the psychiatrists don't really use this antidepressant anymore because they've moved on to better antidepressants. Now, this one had some annoying side effects. Nothing as bad as having lung cancer, though, okay? And this antidepressant is melting the tumors. Now, this is neat. This is the 6th one that's worked for us, okay? We got transplant drugs. We got a lot of these things coming out. Why am I proud of this one? Because we went from computational prediction. We got it working cell lines in a dish. I'm not even going to show you that. We got the mouse model working. 15 months later, after the computation, we got IRB approval, and now we have 2 patients already recruited at Stanford on this drug. And this is the new world for us as computational scientists, as informatics folks. Why do we keep thinking we're handing this off to someone? Most of the time, it has to come down to us to run with our ideas, right? And nothing is stopping us from doing this. 15 months, hell, I think we could do it way faster. Now that we know this is our new finish line, right? For us, we thought, oh, we got to work in a mouse. That's a great finish line. No. The real finish line is to get it to patients, right? And this, how much does this trial cost? We got an internal seed grant, Cancer Center seed grant, $25,000 grant because it's already in the pharmacy here, right? Sometimes the clinical trial isn't $1 billion or even $1 million. Maybe that seed clinical trial only costs a couple $1,000s, and your institution might already be ready to give you money to run with your ideas. The point is that it doesn't end with the computer. It might not even end with the mouse. It might really end. The reason we are all in medical schools is to do something about it with patients and healthcare providers here. So we have 10 such stories coming like this. You're going to see the papers. In fact, my usual rotation project now for a grad student is go figure out a drug for this disease in 10 weeks. If I like what you do, maybe we'll keep you in the lab, okay? But that's what a Genentech would spend, I guess, 6 years doing. That's what they got to do in 10 weeks now, right? I think a kid can do this today, especially a grad student, maybe even an undergrad here. So I've only got 5 minutes left, so I'm going to just end with my big picture, okay? Why systems medicine, where I think this field is going, and I'll leave a couple minutes for questions here. When I think about fast forward where all the problems are in your health system and mine and all across the world, it comes down to patients and patient care with chronic disease in the end, okay? Why I go after cancer is because it's becoming a chronic disease, diabetes is chronic disease, obesity. Most of my field of pediatrics is disappearing weekly, right? We don't have kids with meningitis anymore. We way more care about kids that are obese because they're going to get this adult disease later, right? Chronic disease is all about it. We have these patients, we want them to head towards good outcomes. Here's a patient heading towards a good outcome. But once in a while they diverge and they head towards a poor outcome. And we only get to see them every once in a while, whether it's at a clinic or the exam room, what I'm calling biomedical moments, right? We don't get to see them. We don't live with them. The circles maybe are getting closer because we might be tracking them with mobile health and patient portals. We only get to see them once in a while, and once in a while we just want to keep them heading towards a good outcome, but we once in a while see they're heading towards a poor outcome. Now the whole entire country, entire world is focused on what in the patient's genetics and genomics is going to keep this patient heading towards a good outcome instead of a poor outcome. But you know what? I'm going to be the first to admit, we can have an entire human genome sequence on a patient. If they can't afford the meds, they're going to head towards a poor outcome. I don't care how many base pairs you have. If they live in a field next to a pesticide, they're going to get a problem, no matter how many base pairs you've sequenced. What in the social environment also alters this outcome? But actually, you know what? You can have the entire genome sequenced on a patient. You can make sure every patient can afford the meds they deserve. Someday we'll get there in the United States. But if we can't practice quality care, these two don't matter. If we don't even have our act together in that biomedical moment to keep them in the right direction, to get a hemoglobin A1C on a diabetic and all the rest, they're still going to head towards a poor outcome. Now, why am I mentioning it this way? Because we have genetics and genomics, we have social and environment, public health, we have quality of care. Each one of these is incredibly data-driven today, meaning that we at least have measurements in these fields. But these are some of the most siloed organizations today. How often does the geneticist really talk to the behavioral scientist, talk to the QI person in the hospital? But actually, their mission is all the same. They're all trying to help this patient head towards a good outcome. And to me, the future is being able to learn from every patient, apply this to every patient from each of these three arrows, earlier in real time. It's what the government would call a learning healthcare system. It's what I would call a data-driven systems medicine approach. So take-home points here, molecular, clinical, epidemiological data and tools already exist. They're there for you to play with. Integrating across data is incredible. Bioinformatics has got to be more than just building tools and websites. The world doesn't need yet another website. If you think you have the best killer app, the best killer algorithm and method, show the world what you found with it, not just the URL for your website. If you built your tool, you know it best. Show people what you find with it. But this is the hardest part, is how do we find new investigators who are willing to launch their career on other people's data? It's a hard story. I don't trust any one of these data sets, okay? But I trust what's the wisdom of the crowd across these scientists, and today we're enabled to do this. Always looking for postdocs. I couldn't have done any of this with a long list of collaborators. I have endowment support from the Lucille Packard Foundation for Children's Health. My lab has 17 NIH grants from these 9 institutes of NIH. These 4 give me more. These 5 give me less. I still love them. March of Dimes, HP, HHMI, cancer foundations. I thank my admin tech staff all the time. I'd never get a grant or paper out the door without them. I always thank my wife, my molecular biologist, biochemist, reads all the big papers and grants that comes out of my lab, starts companies with me now, and lets me go all over the place to give talks like this. Thank you very much. I think I left a couple minutes for questions. I won't be offended if you run out the room.
Yeah, thanks for a very interesting and enlightening talk. What I'd like to do is, when people, we have a few minutes for questions, is hand you the microphone because we're recording this, so that way we'll capture the questions. So let me run up to Susie and
So I don't know how to ask this, but what
Sure.
do your grant applications look like, and how do you
What do my grant
avoid the
applications look like?
fishing expedition problem?
I'll tell you how exactly my grants look like, okay? So, first of all, you have to be, there's a huge element of grantsmanship involved here, okay? But my grants typically look like, aim one is, look at this cool computational method I have working, like here are my papers. Aim one, I'm going to make it better. Aim two, I'm going to prove to you why it's working better with evaluation in silico, let's say, this data set, that data set, compared to the other methods. And aim three, here's real biology I'm going to run with this method, okay? And I might get collaborators who've signed they're going to run with anything I predict, this disease or that disease, or increasingly I might just say, we've got a company, we might have found them with Assay Depot, I might not even mention Assay Depot, I'm just going to quote in there saying, this company is ready to run with this experiment. Now, you got to be obviously cognizant of what's going on here, right? If I say I'm going to Assay Depot this mouse experiment, send this grant to a mouse study section, they will laugh me out of the room, okay? I grant you that, right? But if you send a grant to an LM or computational study section, well, again, I have to think, all of a sudden they're seeing someone that actually wants to try this idea, right? Instead of just leaving it for someone else, the great unknown people to hit a website, you're actually going to try this idea? It seems to work for me. Seems to work for study sections. So it means sharing some of the money, okay? Not just take it all for you as a junior faculty. Share it with experts who are actually there to get your success just as much as you want your success. Does that answer your question?
Thank you. Any other questions?
What types of skills are people coming into this field needing?
Yeah, what skills are people needing? So it's a complicated question. So for the molecular side, so what it really comes down to is we need more quantitative thinkers in our field, okay? And what often that means is people sometimes go into biology and medicine because at least in the old days, they can't stand the quantitative engineering side of things, right? The reason why I keep talking about high school students, right, is just to show people, look how fun it can be learning biology in high school when you look at 30,000 breast cancer samples instead of dissecting frogs, right, with formaldehyde. That's not biology. It doesn't have to be biology. It could be as cool as finding a new diagnostic and patenting it as a high school kid, right? So it means keeping the kids, and by kids I mean at all age, keep them as quantitative as thinkers as far as they can go, right? So empirically what it means is today, when I look at our incoming applicants for our graduate programs, a lot of them are undergraduate majors in biocomputation. There are many undergrad campuses that have this. Certainly Stanford, Brown, where I went to school, UCSC, UCSD. Many campuses are now thinking or building computational biology programs. It could be housed in the Department of Computer Science. It could be housed in the Department of Biology. It doesn't really matter. But there's students that are getting some dual training, and they're going to grad school. So those are going to be those applicants. Some of them go to medical school. They're incredible. You know, computational biology undergrad, to get into medical school is just an incredible story for them. I've seen students where multiple institutions are trying to woo them to come with fully funded MSTPs, right? You get an MD PhD from our program, we'll fully fund you, and they get multiple offers like that on arrival on campus, not at the end of the day. Like, they're really getting sought after. So try to get undergrads interested in this stuff. I know you guys don't have undergrads on your campus here, but you have them in your community. Maybe that's your pool. Maybe that's your pool of future grad students. Maybe it's your pool of innovators to start the companies with you guys too. Thank you very much for having me here.